All right, everybody, let's get started. It wouldn't be UCLA fashion if we weren't a little bit late getting started. Okay, so welcome. Well, it's short, so I gotta adjust it. Welcome, friends and family, to our 2023 Nanoscale Microscopy final presentations. We're here at the California Nanosystems Institute at UCLA. My name is Dr. Shailene Tanimoto, and I'm the education manager here at the CNSI, and I've had the pleasure of working with your students and watching them all week learn and grow. And we're really excited today to show you all that they've learned. And so I'd first like to welcome you all to UCLA. UCLA was founded in 1919, so 104 years ago. It's the second oldest school in the UC system. And our campus spans 419 acres. And so it's a big campus, and the students had to walk a lot this week. And I'm sure you might hear about some of those complaints. There's lots of hills too. And I think they've become experts at finding elevators around campus so that they can get around and avoid those hills. So UCLA offers 337 undergraduate and graduate degree programs. We have 31,600 undergraduate students. 10% of those students are international students. 31% are first-generation undergraduate students. And 42% of undergraduates are majors in the life and physical sciences. And so we're here today at the California Nanosystems Institute, or the CNSI. The CNSI is one of Governor Gray Davis's institutes for science innovation, and we're here at UCLA. The CNSI was originally established in 2001 as an engine for innovation based on the belief that the defining technologies of the 21st century will arise from understanding and constructing functional nanosystems through controlled integration of nanoscale components. And so our mission is to be a world-leading nexus of research, education, and translational activities by engaging in scientific discovery that fuels innovation and impacts society. And so CNSI education is led by our Director of Education, Dr. Rita Blake, me, your education manager, and our program coordinator, Dr. Elaine Marita. And, you know, we are a department here at UCLA, but we're not a degree granting department. Unfortunately, you can't, at this time, major in nanoscience here at UCLA. But instead, we are an interdisciplinary hub of research, technology, education, and entrepreneurship. Our education outreach programs include K-12 teacher training workshops in nanoscience experiments, a nanovation competition for 6th through 12th grade students, public outreach events, and these summer session programs for high school students. So this nanoscale microscopy course is one of our summer sessions programs, and it really combines this research, technology, and education. And so here at the CNSI, we have a variety of tech centers, and they all have names that are a mouthful and crazy acronyms. Again, a UCLA tradition. But this course specifically focuses on these 3 technology centers. So the Electron Imaging Center for Nanomachines, EICN, those are electron microscopes. The Advanced Light Microscopy and Spectroscopy Lab, the ALMS, those are light and fluorescence microscopes. And the Nano and Pico Characterization Lab, which are scanning probe microscopes. And you'll hear a lot about those different microscopy techniques today in your student presentations. Okay, so this was our course schedule. And as you can see, over the last 4 days, your students have had very jam-packed, rigorous content. Our learning objectives for this class are not just in science, but in our skills in science that we did want them to learn is about nanoscience, about microscopy, and about being a scientist. But we also wanted your students to have skills in life. And so it's really important to focus on a growth mindset, this belief that a person has that they can learn more or be good at anything if they work hard and persevere. We also emphasize critical thinking, communication, collaboration, creativity, curiosity, problem solving, and reflection and introspection. So we hope your students demonstrate these qualities beyond our classroom. And you know, a lot of amazing people helped to make this program happen, and we'd just like to take a moment to thank some of them before the presentations by your students. So first, we'd like to thank our nanoscale microscopy instructors, Dr. Laron Bentalila, the director of the ALMS, Dr. Adam Stieg, our director of the Nano Pico Characterization Lab and director of Technology Centers here at CNSI, Dr. Hong Zhou, who is a professor in the Department of Microbiology, Immunology, and Molecular Genetics, MIMG, again, another acronym that's way too long, and Dr. Matthew Mecklenburg, who's the managing director of the EICN. So thank you to our instructors. We want to applaud them. And on Monday, we had some amazing guest lecturers who inspired some of our students with their research projects that they'll present. So Professor Yuzheng Li from the Department of Chemical and Biomolecular Engineering, Professor Dolores Bozovic from the Department of Physics and Astronomy, Professor Jeff Miller from the Department of MIMG and the CNSI Director, and B.G. Bai from Professor Ozken's lab in the Department of Electrical and Computational Engineering. So thank you. Okay, we also had teaching assistants, resident assistants, and technology center staff help us this week. So our teaching assistant, Diane King, our resident assistants, Ethan Chan, Kylie Lindgren, and Holly Cinnabandith, and our technology center staff, Haley, Brian, Evo, Alexander, Sarang, Paul, and Michael. Thank you all. Okay, and last but not least, we want to thank our CNSI staff. Lucas, our IT manager, helped us get a bunch of USB to USB-C connectors so students could use the microscopes. Our digital communications manager, Mark, in the booth in the back, is helping us live stream this event. And he'll make you all look really great on camera on the YouTube forever. Our events manager, Mitch, and our work study students, they helped get the food for you all week. So thank you to you guys. All right, so now it is time for the final project presentations. So the students are working in groups of 3, and they will present a novel research project idea using a nanoscale microscopy technique that they learned this week. They'll present their idea for about 7 minutes, and then we'll have 3 minutes for questions from the audience. We highly encourage questions from family and friends so you can better understand what your students learned this week. We will have the first 5 groups present, then take a 10-minute break and resume with the final 5 groups. So this is our schedule here. In case you're watching later and want to jump to the section with your student, you can see where they're at. Okay, so we're going to start with group 1. Come on up and we'll get your slides up.
Hello everyone, and welcome to our presentation titled Using Porous Casing to Decrease the Weight of Lithium-Ion Automotive Batteries.
My name is Isabel Quinones.
My name is Karina Parks.
And my name is Sarah Leisman.
Okay, so starting with our abstract. Lithium-ion batteries make electric vehicles extremely heavy compared to their gas counterparts. I don't think it's on. Okay, I don't know. Lithium-ion batteries make electric vehicles extremely heavy compared to their gas counterparts. This poses a substantial risk to lightweight gas cars on the road during a collision. To lessen the weight of these the weight gap between the electric and gas vehicles, we plan on creating a lighter lithium-ion battery. To do this, we will implement pores in the aluminum and silk battery casing, which will decrease the overall battery weight. To find the optimal metal casing porosity, we will use scanning probe microscopy. This technique will allow us to find the porous material with the highest porosity possible while still successfully containing the battery.
We will be utilizing the technique of scanning probe microscopy in our project. This technique fills the specimen with a sharp cantilever and generates information on the surface of the sample. You can see 2 images on the left-hand side of scanning probe microscopy. We will go into more detail into how this works later in our presentation. This research concept will aid the field of battery technology research by allowing lighter batteries to be produced with the same energy density as regular electric batteries. We'll be focusing on lithium-ion batteries, as you can see on the right.
Now I'll be going into the precedents of our research topic and giving some background info. This past Monday, we attended 4 guest lectures regarding different applications of microscopy. The first of these lectures was on recent advancements made to the function of lithium-ion batteries. Prior to the lecture, our group was aware of the safety risks posed by electric vehicles. The immense weight of electric batteries in vehicles poses a substantial liability to light gas cars on the road. If a heavy electric vehicle collides with a gas vehicle, the latter will be significantly more damaged. Current scientists are approaching this weight issue using a number of techniques, such as with nanotubes or solid-state batteries, which can be seen on the left. Like the scientists, we initially researched new materials to include, but found that the current lithium is the most optimal material due to it being lightweight and having a high energy density. So, instead of changing out the lithium, we looked into how we could manipulate the battery's casing. In fact, around 60 to 75% of an electric vehicle's battery weight comes from the individual cells, while the remaining 25 to 40% is made up of the battery's casing and management systems. Therefore, by decreasing the weight of the battery casing, we could potentially decrease the entire battery's weight by over 25%. Here are 2 tables comparing the weights of electric cars and gas cars. I used information from cars with comparable sizes, so it's easy to visualize how much the battery contributes to the overall weight of an electric car. You can see that the batteries on the left-hand side contribute to about 1/3 of the electric car's total weight, while the engines are only about 1/10 of the gas car's weight.
The microscopy technique we are applying to our idea is scanning probe microscopy. This implements the sense of touch to learn more about a sample. As you can see in the top image, the microscope works by a force applied to a spring-like cantilever when the specimen and cantilever are close together, which causes the cantilever to bend. This changes a laser's reflective angle, which is captured by the detector. The cantilever tip has a single atom, which is there because there is always one atom that sticks out a bit farther than the rest. Images are later generated from essentially a data set of coordinates, which are translated into a surface image. From this technique, we can see the surface of the battery in atomic resolution, so in great detail. Below is an image of a scanning probe microscope, which does look quite different from your average light microscope. Current scientists are trying to solve the problem of making automotive batteries lighter, and scanning probe's high surface magnification can assist in these efforts.
Okay, so now moving on to our solution. What are you proposing? After realizing that lithium is the most energy dense, efficient, and lightweight element to use for batteries, we instead opted to improve the aluminum and steel casing on the outside. We plan on substituting porous metals for the outer casing of the battery. Taking some inspiration from the process of inverse opal, which is the process of scooping small holes out of the desired material, we decided to implement a similar process in our project by creating a porous casing for the outside of the batteries. By adding these pores to the aluminum and steel casing, we'll be able to remove excess mass from it and therefore making it lighter. But how do we specifically plan on doing this? By making these pores, we would involve using a gas from a pressurized atmosphere, such as hydrogen. And these pores are made for when the insoluble gas, when the molten metal dissolving the gas becomes solidified.
As Serena stated, electric car batteries are encased in steel or aluminum to protect them from mechanical damage and provide chemical resistance. As stated earlier, however, this metal casing can contribute to up to 40% of the battery's total weight. Porous aluminum is a type of aluminum with a porosity of around 55 to 65%, and it's a high specific surface area. Porous aluminum and other porous metals, including steel, have been used in shock and sound absorption, thermal insulation, heat dissipation, and for fabricating membranes and filters for bioremediation. Substituting the current metal battery casing with porous casing of the same metals will therefore decrease the battery's weight but maintain its function. In order to ensure how high of a porosity of the metals to substitute, we plan on using scanning probe microscopy, or SPM. This type of microscopy will allow us to examine the porous metal on an atomic level. SPM would enable us to measure the forces between the probe and the surface of the metal. We would scan surfaces of metals with different levels of porosity after time spent encasing the battery, and we would then compare it with a control casing. The ideal metal porosity would be the highest possible porosity without degradation from the encased battery. Our experiment would not be possible without SPM because it is crucial that we use data from the metal surface to determine the highest porosity possible.
Now, you may have the question of, will creating these holes really decrease the weight of lithium-ion batteries? By making the casing more porous, we were removing some of the weight rather than simply pushing the material downwards to make holes. This would account for a decent amount of material removed from the casing overall. And although this method focuses more on improving the battery's exterior rather than its interior, the solution for the future can be improved and uses an invitation for future improvement on this idea, and possibly the idea of using this limiting material weight onto other scientific areas. A few other questions that may pose for discussion are, once the optimal porosity level is achieved, how can you further improve the weight? Are there any other microscopy techniques that we can use to observe this porous aluminum and steel case? And are there any other replaceable materials besides aluminum and steel?
These are some of our references, and we would like to thank everyone involved in educating us at CNSI.
All right, so any questions from our audience? Start with Dr. Rita Blake.
So my question actually references one of the future directions that you pointed out, which was something I wanted to ask anyway. So you mentioned that there may be other techniques that you could use to image the pores. So my question is, why would you want to start with scanning probes specifically and not, for example, electron microscopy, which can also show you what the pores look like?
Okay, so for scanning probe microscopy, we're able to specifically analyze the surface of the casing. So we're able to really compare the specific surfaces of a control surface to the one that is that we're testing out. So by using the tip, the touch mechanism of scanning probe microscopy, we're really able to feel how the holes are gonna work for the battery, if that makes sense.
Other questions?
So how are you planning, or how would you plan on testing the how the porosity affects the battery's performance? Once you've bored the holes, how are you gonna test how well the battery works, or how do you make sure that it hasn't messed up the battery internally?
I think the main thing we're going to be testing is if the battery has has any leaks coming out of it. But for future research, we will be seeing the performance of the battery, but the main thing we're focusing on if just the structural integrity has been changed. Do you have anything to add?
Yeah, adding on to Isabel, we plan on testing the aluminum and steel porous casing at different levels of porosity, so maybe 20% or 60%, and trying different levels of porosity and then comparing that to an initial porous casing that hasn't been affected by a battery. And we can find the highest porosity without any leakage or visible, well, not visible, but without any damage from the battery.
Any last questions? Okay, great job. Okay, can we get group 2 up here?
Remember, we have this with our laser pointer.
Hello, we're the CNSI Nanoscale Microscopy Summer Institute 2023, and we are going to present about how artificial intelligence could improve the process of overlay correlative light in electron microscope image. I'm Juwon from FPS, and she's Angela Pig from SSI. She's Anna Pig from BC College 8. Before we begin, I'm going to summarize our research. So our main question is, how can artificial intelligence improve correlative confocal and electron microscopy by overlaying them? So we're going to compare images produced by confocal light microscope and electron microscope to achieve results by leveraging AI to improve the process of overlaying CLEM images. This offers benefits in terms of efficiency, accuracy, standardization, advanced analysis, and the potential for new discoveries. It empowers researchers to delve deeper into the microscopic world and expand our understanding of biological systems.
For next, I'll talk about concept and topic. For confocal microscope, it is high definition imaging using a light to visualize the structure of molecules within cells and tissue. And for the electron microscopy, it is a high definition imaging using electron beam to visualize subcellular details of cells and tissues such as membranes and organelles. So why scientists overlay CLEM image? Scientists overlay these two microscopic images to combine the benefit of both technique. Confocal provide the information about the molecule or structure that labeled with markers, while the electron offer detail at high definition. So by overlaying the image, scientists can clearly complement the fluorescently labeled structure with their subcellular context. Also for comprehensive understanding of cellular system and function.
So I'm going to talk about the precedent. So before the application of AI in the process of overlaying CLEM images, the alignment and registration of light microscope and electron microscope images were primarily performed manually by researchers. So this manual approach involved visually comparing and aligning the images. However, this process was time-consuming and it was prone to human error.
For the next precedents, I'll explain about fundamental of two microscopes. First, component microscope is a high-resolution imaging technique by scanning different planes and blocking out of focus light. As you see the components of microscopy, a focus layer beam and pinhole aperture selectively eliminate a single plane of specimen. The emitted light is detected by a sensitive detector, and computer software reconstructs the acquired data into three-dimensional images, as you see in the bottom of the slide. Next one is electron microscopy. It is powerful techniques that utilize a beam of electrons to visualize the nanoscale structure of samples at high magnification and resolution. An electron source generates a stream of high voltage electrons, and a beam of electron is focused onto sample, and this interaction scatters signals collected through a detector and transport into an image. Next one is our solution, and from the solution, we propose an optimized method for overlaying cellular structure for electrons and light microscopy by using AI. So how does it work for our solution? First, ensure proper alignment of microscope's image and adjust the magnification of each image so that identical structure appears at the same size. And next, orientate microscopy's image and overlay to highlight the ultrastructural features. By using AI, you can skip the complicated steps to overlay, like using Photoshops. So why is it better to use this solution rather than using original one? Is first, save times and labor for expert, and reduces cost for training and kind of installation of Photoshops, and minimize variation in staining quality, and lastly, it can eliminate human error throughout overlay. Lastly, I'll talk about our future direction. So how can the AI also help the process before overlaying the image? When we see the first step of our solution, scientists need to show a proper sequence of image and modify the magnification of his image, but we can use the AI tools to do this work and make less job to do. And for second question is what development can made in usage of AI with real-time imaging system. So developing AI real-time imaging system can make quick feedback and modification during test and develop the control and gaining the data.
These are our references, these are our contributions, and thank you for listening.
Okay, questions from the audience, please.
So, how are you planning to train the AI to do this, to synthesize these two images? How are you planning to have the AI put them together?
So we proposed this solution by the lecture from guests, but we are not sure how to use AI. Yeah.
So I guess like this technique, this correlative light electron microscopy technique exists, right? And people do this by hand already. And so in the AI, could you go back to the slide where you have the AI, the training model, right? And so people already acquire these images, correct? Like these light microscope images and these electron microscope images, right? And you're saying that by hand they used to do all of this where they combine them and then they end up with a combined image, right? So your ground truth in this case is what people have already done, right? And then they have the before images of the two separate images before they're combined, correct? Yeah. And so you would train the model, right, with the stuff that's already out there, where they already do this CLEM. And then you train the model, and then hopefully your model is good enough so that when you give it new images of the light and the electron microscopy, it could then put it into this never before combined image, correct? Yeah, so that's okay. Any other questions from the audience? I am also not an expert in AI, so it's kind of like computers do this thinking stuff for us. Yes, Dr. Rita Blake.
I guess normally this is done with confocal and electron microscopy, and I'm just curious if it can be done with any two imaging techniques that you could overlay with each other. Like, is there a reason why it's always confocal and electron microscopy? Like, could you do it with, let's say, like, I don't know, confocal and scanning probe, or scanning probe and scanning electron microscopy?
I believe that it can be done with any two kind of images because it's just using the AI technique to overlay images that are already there.
Very interesting presentation. I wanted to ask you maybe take a step back and maybe if you could explain why it is important in the first place to merge images from two different techniques. What is it that you want to gain from that?
To use the benefit of like both like a technique and some like advantage of both Microsoft and yeah.
So what do you learn from the light microscopy? What do you learn from the electron microscopy? And what do you gain from putting them together?
By overlaying the image, a scientist can get a clear complement the fluorescently like a label structure.
And that's from the fluorescence?
Yes.
And then what do you get from the electron microscopy?
Mm. For from the electron microscopy, we can get like really great detail of like high resolution of the image. Yes, and that can combine into one simple structure of image.
So just to summarize, right, you were saying that with the fluorescence microscopy, you only see things that are fluorescently labeled, right? And then in the electron microscopy, you're getting kind of scattering from the whole sample. And then you can combine those two things to get what you're talking about, a comprehensive understanding of cellular organization and function. Yeah? Good? Okay. Great. Thank you guys so much. Okay. Group 3, please.
Hi everyone, my name is Lauren Weiner and I'm from Xavier College Preparatory.
I'm Gleb Bakatilov and I'm from Institute of the Rosenberg.
And I'm Natalie Lane from Los Gatos High School. Our project is on optical microscopy and cellular AI. Abstract. Optical microscopy is a fundamental pillar of microscopy. For our project, we want to look into utilizing fluorescent microscopy in everyday life. Something most people own is a cell phone. It would be useful to create a phone case that enhances your phone camera into a fluorescent microscope. This could help with disease outbreaks in remote areas. Currently, in remote areas, it can take days to get samples in the lab and looked at under a microscope. By the time it is analyzed, it may be too late. Combining precise fluorescent microscopy with artificial intelligence could be revolutionary. It would help remove the need for a doctor to analyze samples. Instead, AI would analyze them at a much faster rate. We want to explore how technology and artificial intelligence can be used in microscopy, making it much more accessible to the world. One of the most important types of microscopy is fluorescent microscopy. Light microscopy is about deriving an image from interactions of light with matter. The human eye can only see a small part of the electromagnetic spectrum. Image 1 shows the electromagnetic spectrum. The image shows that light microscopy is able to pick up very small particles at the nanoscale that the human eye cannot see. In optical microscopy, we utilize many different wavelengths and energy levels to image our sample. In this type of microscopy, lasers are commonly used to provide a more direct beam of photons and to concentrate the light. They are most commonly used in fluorescent microscopy. In fluorescent microscopy, a high-intensity light source is used in order to excite a fluorescent species. Images 1 and 2 show the fundamental parts of a fluorescent microscope and how the light is reflected and refined. Creating a technological device to enhance a phone camera would turn a phone into a mini microscope. Then, with the addition of artificial intelligence to help interpret the images, it would greatly advance the medical field. In remote areas, doctors with microscopes are hard to access. With our cell phone case addition, the doctor would be taken out of the picture. Instead, AI would read the results. In turn, this would allow for doctors to spend time on other patients instead of traveling to remote areas. It would make microscopy much more accessible to the world.
So I'm going to be going over the precedents. So right now, the current state of optical microscopy is limited in the resolution and depth. And on the other hand, fluorescent microscopy is a more powerful technique since it provides sharper images, improved resolution, and 3D models. However, it's limited to specialized lab due to expensive equipment and skilled operators. And the manual analysis of microscopy images also poses delays and requires expertise. Therefore, before our proposed solution, accessibility and efficient analysis were challenging in remote areas or time-critical scenarios. The fundamentals of fluorescent microscopy in combination with AI. Our proposed advanced microscope technique brings smartphone integration and AI analysis into play. We believe with the help of smartphone integration, we can make fluorescent microscopy accessible to a wider audience. The proposed technique involves utilizing a smartphone's camera and using a specialized phone case or attachment equipped with additional optics to transform the phone's camera into a fluorescent microscope. This setup would allow users to capture high-resolution images or videos of their samples directly using their smartphones. Furthermore, AI analysis will enhance the efficiency of the smartphone integration. Captured images can be processed and analyzed using AI algorithms, since AI has demonstrated remarkable capabilities in image recognition and pattern detection. By training AI models on large data sets of microscopic images, the system can learn to identify specific cellular structures, pathogens, or abnormal patterns associated with diseases. This would enable real-time analysis and identification of samples without the need for a human expert on site.
All right, moving on to our solution and a bit of a summary about what all of that was. First off, I'd like to read this quote from the OSCAN Idogan Research Group. This cell phone-enabled optofluidic imaging flow cytometer could especially be useful for rapid and sensitive imaging of bodily fluids for conducting various cell counts. And one example is monitoring of HIV-positive patients or rare cell analysis, as well as for screening of water quality in remote and resource-poor settings. So, how does this work? These pieces of technology would have the ability to convert phone cameras into fluorescent microscopes that would utilize AI samples. Talking about how AI works with these samples, the sample would be collected. Would be collected, and the AI would be trained on how to read these samples and then produce the diagnosis to the patient. So why is it better than taking a sample out in rural areas, sending it to a lab? It's a better alternative because it doesn't require the medical professionals, and having these people in the rural areas is expensive and extremely time-consuming. So some future directions and implications of this project that we've proposed are: how much will this cost, and will these costs outweigh the traditional costs of performing such procedures? We hope that these procedures and these costs will be covered by grants. And what kind of people would be able to make this product? We would rely on nanomicroscopic researchers to make the phone attachment, and then specialized tech industry people to formulate the AI inside of them. So what are the limitations of this product? It still will cost money, and it will take time to build and train the AI, not just for bodily fluids and samples, but also water samples, and to analyze them and come back with not just images, but also a direct diagnosis. Future direction of this would be three main things: availability, global usage, and less manual fluorescent microscopes. We hope that our project would be taken out of just rural areas and into homelessness and poverty-stricken regions. Thank you for listening to our presentation. Here are our references and our contributions. We would love to hear your questions.
All right, questions from the audience, please.
Hi. First of all, I want to say that I loved your presentation. I thought this was such an ingenious idea, and it was very innovative. My question was, you guys mentioned specifically that your target audience is for residents of rural areas, and at the end you mentioned people who may be impoverished. And I was wondering if you think that these people would have access to the cell phones to see the AI images? Like, would they have some sort of access or not?
I believe that there are people going out into these impoverished and homeless-stricken areas, offering help already in clinical vans and whatnot, offering pillows, blankets, and normal testing. We believe that these devices would be able to be taken into those organizations and then given to the homeless and poverty-stricken people for free of charge. I hope that answered your question.
So my question is about the samples that you're imaging. So so my understanding is that typically with fluorescence microscopy, you need to have a sample that fluoresces, right? So how are you collecting samples and how are you prepping them for the fluorescence microscope that's on the phone? Like, is there not any sort of like tagging or fluorescent markers that you need to do? Are you relying on only imaging things that fluoresce kind of naturally? Like how are you actually going from collecting a sample to being able to image it?
I believe that we thought about this project and fluorescence would be added to our samples in some type of way. And do you guys want to add anything? So we just planned on adding some type of fluorescent organism or coloring to our samples.
So then I guess as like a sort of, I suppose a follow-up comment more than a question. Much like you were talking about with people sort of aiding people experiencing, homelessness or other sorts of issues like that, I think when you're saying like it's eliminating the need for a doctor, that might not be so much the case, but what it does do is maybe eliminate the need for a big fancy lab that they would have to go to that's far away. So I might rethink this not necessarily as something that eliminates the medical professional, but something that makes their job more portable and therefore easier and faster in that respect.
Other question?
Very exciting ideas, actually. I'm just going to add maybe a little comment to what Rita just said. I don't think we can eliminate the doctor, right? But I think really with that great idea, what you can eliminate is actually transporting the sample. Because I think, imagine yourself in the middle of the jungle, right? And you are taking a fluid of any sort from a patient. What is the most critical part of the process here is to keep the sample viable, right? And usually what you need the most is ice. So just the fact that you don't need ice, right, and you can actually look at your sample right on the spot, I think will be fantastic. So thank you for these ideas.
One more question.
So for as was mentioned earlier, for fluorescence microscopy, you need like GFPs or green fluorescent proteins or something, but you also need like a laser to, to cause the thing to fluoresce. So are you planning on like attaching like a little laser to the phone thing to like how are you going to make them fluoresce?
Yeah, so we're planning to incorporate a laser into the attachment so that it will make the sample fluoresce.
I think it's also important to note that these would be under 200 grams, so it would be a smaller laser, and we would be taking smaller samples.
Okay, thank you. Okay, group 4, please.
Good morning. Today we'll be talking about using light sheet microscopy to study the contraction of fluorescence. I'm Rachel from Ayala High School.
I'm Lucy Park from Whitney High School.
I'm Anya from Doherty Valley High School. So first, our abstract. Antibiotic resistance is a problem that is becoming increasingly prevalent, and solutions are needed. One proposed solution is using pyosins, contractile nanomachines that contract and inject themselves into and kill bacterial cells, as an alternative for antibiotics. Scientists have studied pyosins using cryo-electron microscopy, and with that have been able to work out the mechanism of the injection process, but don't know the details of it. We propose to use light sheet microscopy to further study them. By generating a video of the mechanism, scientists could better understand the process and apply the understanding to the development of pyosins as a new form of medicine.
So now we will be going over the concept and topic. The microscopy technique that we'll be covering is light sheet microscopy. Light sheet microscope will be effective when studying the contraction of pyosin because they normally use thin sheets of light to image samples. Therefore, compared to other microscopy techniques, using light sheet microscope will be better to view the mechanics of the contraction of the pyosin. The microscopy area that will be involved is light microscopy, and the scientific field that the topic can help is the medicine and biology field. With the further research related to pyosin, there can be more medications created which contributes to the betterment of social health.
So previously, scientists used cryo-electron microscopy to generate 2D images of before and after the pyosin injects itself into the cell. And then using these, they created 3D models that they then used to figure out the mechanism of the process. And what they found is the pyosin is made of two hollow tubes, the outer tube called the sheath and the inner tube. And when the pyosin attaches itself onto the cell, the outer tube contracts and pushes the inner tube into the cell membrane of the bacterial cell. The inner tube, which is then lodged into the cell membrane, acts as an ion channel that destroys the membrane potential of the cell, killing it. But again, all of this is based off of 2D images of before and after the injection, and the scientists don't have any actual videos of the process.
So talking about some of the essential parts of the light sheet microscopy, we have cylindrical lens, illumination objective lens, detection objective lens, fluorescence filter, and camera. And the sample will be placed over here. And in terms of their functions, light sheet microscopy uses the light sheets, whereas other light microscopy like confocal instead uses beams. This allows the imaging of the sample movement. The illumination beam is perpendicular to the detection axes. As you can see, this 90 degrees. And this minimizes out of focus light. Light sheet microscopy also uses a cylindrical shaped lens that only allows very thin volumes to be illuminated, which means that there is minimal potential damage to the sample. The sample is prepared on a movable stage, making it possible to scan across the large samples and also view from multiple angles. So what we're proposing is to use light sheet microscopy to capture the mechanism of the contraction of pyosin.
So how would this work? We would use the light sheet microscope to image the pyosin injecting itself into the cell. So we would make a sample that consists of a bacteria cell and add pyosins to it. And it would create a video because if it's high frame rate. And basically what high frame rate means is it can take a lot of photos in a short amount of time, which means that the video creates will be very smooth, and it would allow it to capture the entire process of the contraction in great detail. And scientists would then be able to study that video and to understand the mechanics of the process better and be able to apply it to future medicine.
So as explained, why is it better? As explained, cryo-EM gives two-dimensional images of before and after the contraction. Using the light sheet microscopy would provide four-dimensional video of the sample during the contraction of the pyosin, having dimensions of X, Y, Z, and time. One of the benefits of light sheet microscopy, other than fast 3D imaging and high frame rate, is that it leads to minimum fluorophore bleaching, which happens when a fluorophore permanently loses the ability to fluoresce due to photon induced chemical damage and covalent modification. Using Light sheet microscopy would help future medicine because being able to look into the mechanism of the contraction would allow the scientists to learn more about this process and further help to study the pyosin, which is potential replacement for antibiotics.
So there might be questions about how our project may contribute to future development and direction. Some questions our project raised that helps their discussion includes what other microscopy techniques could be used to learn more things about the pyosins. This question may lead to further research since as technology get more advanced, the microscopy techniques might lead to more faster and efficient ways to learn about pyosins. Another question is that if using light sheet microscopy for pyosins is successful, can we use it on other bacteriophages? This question is significant because if the technique is effective with researches in pyosins, it opens up a lot of possibilities for other bacteriophages research. This topic is able to significantly extend the impact of future development since the successful researches of pyosin using light sheet microscopy is able to develop bacteriophages into medicine to replace antibiotics, which can also help to solve the problems of antibiotic resistance. Researchers are also able to learn more about pyosins and other bacteriophages, which opens up the alternatives that are available for medications when treating other patients. And this is our reference page. Thank you for listening.
All right, questions from the audience, please.
Hello. I really liked the presentation. I have a question similar to the last presentation, which is how do you plan on having the pyosin fluoresce?
So to have it fluoresce, we would kind of just use the standard technique used to fluoresce pyosins or like bacteria cells to put into the fluorescent microscopes.
Great job, ladies. Quick question. So I talked yesterday at the end of my lecture a little bit about how each microscope has advantages and disadvantages and limitations. And obviously, electron microscopy has been used to study the structure of this mechanism, but it has limits, right, which you pointed out. I'm wondering what limitation you think you're going to have to overcome. There is a specific one, and I have in mind about what light sheets can and can't do, and optical microscopes can and can't do with respect to imaging structures that are this size, right? It obviously opens up the time, the dynamics, but what do you have to do to push light sheet microscopes forward to actually be able to do this type of an experiment?
One of the things that we found while we were researching was that for like dual light sheet microscopy, like it will be hard to align the sheets together. That's one of the things that we found. Sorry, I don't know about the other.
So I think he's asking about the size of the pyosin, and it's really small, right? And one of the benefits of using electron microscopy is that the wavelength of the electrons allows you to see things that are smaller, right? And so, you know, what is the light sheet microscopy capable of resolving? And is there potentially another technique that you can use in conjunction with this that would allow you to probe things that are smaller and still get dynamic measurements?
So light sheet microscopy, it's still light microscopy, which means it's not used to image things that are smaller than like bacteria or like cells. So if we did use this technique, the resolution would be something that we would have to improve on. We could use it in conjunction with super resolution light microscopy, which has better resolution, and that could help us improve the resolution and get like a clearer image.
Any other questions from the audience?
I wanted to ask you, while you were researching on pyosins, have you found any indication about the time frame at which that process of entering the bacteria happens? Are we talking about minutes, seconds? Is it like a fast process or is it a long process? Did you find anything in the literature? I'm totally ignorant about that. So.
Yeah, we didn't find that much about it. So far, all we know is that it's a very fast process, probably like a few seconds. But because all that was used to image it was cryo-electron microscopy, they didn't actually like get to see the details of it, so they don't know exactly how long it took.
Any other questions from our audience? Okay, if not, thank you guys. Okay, group 5, please.
Hi everyone and welcome to our presentation. My name is Daniel Park.
I'm Sawyer Karish.
And I'm Jed Mahfouz.
And our research topic is deep learning enabled cancer predictions, unveiling cell transformation through optical microscopy.
So now I'll be giving a general overview of what we intend to address, which is the critical issue of cancer diagnosis through the utilization of the power of optical microscopy to predict the growth of cancerous cells. So early detection of malignant change is critical for effective treatment, but current approaches do frequently struggle to detect the subtle cellular changes that do lead to cancer. So this study seeks to uncover the unknown patterns and distinguishing features linked with cancer development by diving into previous examples of malignant cell production. This study also aims to uncover an innovative approach to cancer prediction using optical microscopy techniques with the potential for improved diagnostics, early treatments, and improved patient outcomes. So now I'll also be giving an overview of the concept and topic that we intend to cover. So optical microscopy is a fundamental tool in the field of microscopy that utilizes visible light to examine and magnify specimens for more detailed observation. It involves the use of lenses and light sources to illuminate the sample, allowing scientists and researchers to study its structure and properties at the microscopic level. By exploiting the interaction of light with the specimen, optical microscopy enables the visualization of cells, tissues, microorganisms, and various materials with higher resolution and contrast. This technique includes several types of microscopy, including bright field, phase contrast, fluorescence, and confocal microscopy, with each offering unique advantages for their specific applications. Optical microscopy has played a pivotal role, especially in medicine, to enable researchers to decode the complexity of the nanoscale world within patients and unearth new insights into the workings of nature. More specifically, through cancer prediction, we aim to investigate how optical microscopy techniques can be utilized and improved upon in order to predict potentially cancerous cells within organisms.
So I'm just going to review current cancer prediction science. We have a few ways of predicting cancer development, the first of which being genetic science. There are some specific genes that code for specific types of cancer, as you can see in the lower image. And that connects to family history. If your family has a history of specific cancers, you may be more susceptible to developing them. And that links to epigenetics, which is how the environment affects your gene representation. So basically, you can be exposed to mutagens, which are basically just chemicals or other things that cause mutations in your cells. You can see them up on the right, radiation, temperature, viruses, bacteria. And there are other risk factors that can increase your chance of developing cancer, such as smoking or a lack of exercise. I'm going to cover the fundamentals of optical microscopy. And it's basically just studying the interaction of light and matter. We shine a light on a specimen. In this case, we use a laser. And we use the reflected light to form an image. Because we're just using light, it's non-toxic, so you can use live specimens, such as the cells we're trying to study. And in order to understand the cell structure, we have to use a specific type of optical microscopy called fluorescence, where we use materials that re-emit light after being exposed to photon energy, such as the green fluorescence protein or quantum dots, which you can see here in the images. SDED, or super resolution microscopy, is a more, it's another type of optical microscopy that uses an extra laser beam to increase the resolution and get better imaging of cells. That's just an overview.
So then the solution we are proposing in terms of cancer prediction involves AI, which stands for artificial intelligence. And as we know, there's been a lot of developments in AI recently. As we know, we all love ChatGPT. And more specifically, the AI we are proposing using is deep learning AI, which is a type of machine learning that uses some fancy computer science stuff, such as artificial neural networks and multi-layer processing. And basically what it does is it analyzes patterns to progressively create higher outputs based off the input that it is given. And we learned about this on Monday through the presentation by BG Bai on how they used the deep learning to produce histological staining. And basically what they did is they used as their ground truth, which is how the deep learning is then programmed, they used a real histological stain as the ground truth, and then they used that to program their model, so then basically what they were able to do was they were able to take a unstained tissue and then virtually stain it by using the deep learning. So we are proposing a similar model in terms of cancer detection. So for us, our ground truth would be we would take a culture of healthy cells just from like your average human being, and then you would, in some of them, you would inject or you would induce cancer. And at all stages of this, you would take optical microscopy shots like Sawyer talked about. So then you would have precancerous cells, cancerous cells, and then non-cancerous cells. And then what we are proposing is that we would train the AI to be able to detect patterns in the cells that have a higher rate of leading to cancer. So our ultimate goal in this is to have an input of a healthy normal the input of a healthy normal cell, just like any one of us, and then the computer would be able to analyze that and output the likelihood of cancer. And this is super important as it provides another way to detect cancer, and it provides another form of potential prevention and just knowledge as in terms of besides just the genetic information Sawyer talked about. So as for the future, one thing that is currently kind of being developed is providing a microscope on top of a phone, which one of the other groups talked about. And this would provide even higher accessibility in terms of cancer because you would then be allowed to just scan. You would take your phone out, put the microscope on it, and then scan your skin, which would pretty much allow anyone who has a phone, which a lot of countries do these days, to be able to check their vulnerability for cancer. I also think this could provide a greater understanding as I think the genetic composition is pretty complex for a lot of people, and I think a 3D model would be easier to understand. And then obviously, eventually our goal is for this to be used to decline cancer mortality rates and contraction rates. So we would like to provide a special thanks to Dr. Tanimoto for putting up with a bunch of high schoolers all week, BJ for originally introducing this topic to us, and then our three proctors, Dr. Bentalila, Dr. Zhao, and Dr. Stieg.
I'm going to provide an additional thanks to our CNSI staff and our RAs.
As well as to all of our guest speakers, Dr. Oskan and the Oskan Lab.
And then these are our references, and thank you for listening. Are there any questions?
Okay, questions from our audience, please.
Yeah. So you guys are, just to make sure I understand it, you guys are using confocal microscopy to image the cells and then put them through an AI to see if they're cancerous. So what about precancerous cells that don't like look cancerous through optical microscopy or other techniques, but are like if you're training the AI to like say like is this cancerous by looking at it, and the AI can't tell just how are you going to avoid like false positives or false negatives where the AI thinks that it has to be able to tell with the cell that you can't tell visually?
I guess my question is similar, which is, what is it looking for, right? Like, what are the features physically that one can observe in an optical image that is an indicator of malignant transformation in a cancer cell, particularly at the early stage, because you want to get it as early as possible? Are there specific things that you know structurally can be elucidated from an optical microscope image that you could train the AI with to be looking for?
So we're using fluorescence, just as a clarification. And so we're looking for like maybe unknown patterns, because this is just a potential research idea. We're going to see if there is something that can be found before it actually develops into a problem. I think Daniel can elaborate on that.
Oh yeah, so in terms of the AI, we don't know exactly what we would want it to look for. That's something that we would have to do further research on, as you know, this is just like something that we're proposing. In terms of the actual like how it would predict it, so how current cancer prediction works is it doesn't say necessarily, oh, you're going to get cancer 100% or you're not going to get cancer. It just gives you a risk assessment. So this would not allow this, like I couldn't be able to look at myself and say I'm 100% going to get cancer or I'm 100% not going to get cancer. What we would want it to do would just be provide a risk assessment of, oh, based off what we're seeing in based off your cellular composition, you have some things that we're seeing that would make it more likely that you may contract cancer in the future or less likely that you would contract cancer in the future.
Can I just say, can you go back to the slide where you had a picture of normal cells and cancer cells? Yeah, so I feel like visually you can tell the morphology of the cells is different, and that one on the right is very clearly cancerous, right? And so hopefully there is a progression from normal on the left to gross looking on the right. And so hopefully that is what the AI is picking up on, those patterns that we might not be able to see with our eyes, but are patterns that arise from looking at a bunch of samples that are non-cancerous, precancerous, and then very aggressively cancerous. And I feel like your thing is like, hey, you might develop cancer, go to the doctor, right? And so it's more about like earlier detection, because once it gets that bad, it's less likely to have good outcomes.
Okay, so first I just want to say I really liked you guys' presentation, but I have a question about the user of this device. So feel like I'm pretty uneducated on cancer research, so let's say the user scans their arm, but the cancer cells are actually in their outer thigh. Would this person have to do a full body scan, or is there a way of detecting it in other parts of the body that the cancer isn't in?
So that's a good question. That's one we kind of like thought about when we were originally trying to think of this idea. As obviously like it's kind of easier to image skin cells as the closest to the top of the surface. So like if you go back, I should have just left it there. But these images are skin cells. So I think the simplest use of this would be in terms of skin cells because they're the easiest to detect as they're on the top of the surface. And if we would want to do other types of cells, I think the answer is yes. We would need to do biopsy to have further depth in that case.
Hello. I really like the presentation. I'm just wondering a little bit about the AI because you're using an image to some form of likelihood of cancer. So how do you think you would train your AI to go from like the image of the cells to directly about like a percentage or some form of indication of the cancer?
Thank you for your question. So, just to answer that, as Daniel also said while he was presenting, we'll be using different stages of the formation from like regular cells as they begin to turn cancerous. So, these will of course be used as ground truths based on past experiences from previous cancer patients, and the AI will essentially compare both types of images to these ground truths and present a prediction as to what these cells may form. This is kind of also answering a previous question that I believe Ellery asked in terms of like anomalies. So these will also be fed to the deep learning AI model in order for it to essentially look at anything that may seem unusual and analyze it in order to see if maybe more normal looking skin cells or any other cells may eventually turn cancerous even if it does not seem so.
All right, I'm going to be the devil advocate, because I think it's totally legit, it's totally valid to use AI and not to expect and not to have any preconception about any pattern to detect, right? Because I think this is the entire point of using AI is to use AI to actually find hidden patterns that a trained pathologist in that case would have missed, right? So I think the beauty of AI would be to actually search for things that we didn't think about or we didn't know about or that are not obvious at all, right? Can you say something about that?
Oh yeah, and I definitely agree with that. I think that's why we don't have like kind of a set list of this is what we wanted to look for to begin with. Obviously, there are things that like there are more obvious things that we could possibly look for, but that's why it is kind of open-ended, right? You know, if we knew exactly how cancer formed, this wouldn't be an issue. And so this is something that humans have struggled with for a really long time, right? So I think that like probably potentially putting a different set of like kind of eyes on it might illuminate different possibilities.
I'd also like to add on to what Daniel just said. So, of course, when you do train a AI model, it tends to generate biases towards more recognizable patterns than like others. So, essentially, if a model is trained too much, it will begin to believe that almost any cell can eventually become cancerous, which of course is the case, but it kind of hinders the prediction abilities that it does have. So you'd want to train it essentially to a certain point where it can still recognize these differences without only doing what any other pathologist would do and going by the standard procedure.
Okay, great. Thank you guys so much. Okay, we are ahead of schedule, which is good, better than being behind. So it is currently 11:17. Why don't we pick up again around 11:30, and we're going to take a biological break, food, water, bathroom, and we'll come back at 11:30. All right? this is this one, and then this button right here.
You in the middle, yeah. One key.
So who wants to be in charge of the laser?
I'll do it.
Press that button, and then you can also advance in the slides like that. And then you have to turn this one back on. But remember to hold it up. You can also use this one too.
I'll just this year. You and Kaylin use those.
Kung ito.
You mag any yours. And you yung laser, you can. I'll do the laser.
Okay, everyone. Actually, my watch says it's 11:29, so we're gonna hold. Okay, maybe we'll just get started. Okay, so we're gonna start up again with group 6. Here they are. So I'm just gonna pass it off.
This. All right. Good morning. Almost noon, everyone. So today for our presentation, our group will be proposing a research idea that we have been planning, and that is to use cryo-electron microscopy to understand gene therapy. My name is Yvonne.
My name's Lorraine.
And my name is Kaylin. Okay, so to give a little bit of background information, gene therapy is when a patient's cells are modified to cure a certain disease. One common cause of gene therapy failure lie on its delivery, specifically when they target failure to target the correct tissue. In light of this, our group's theory is to use the microscopy technique of cryo-EM to analyze the delivery carriers that are used in gene therapy, and so that the structures of the carriers can be modified when researchers use this process and increase the efficiency of gene therapy.
So for the concept here, let us get to know more about gene therapy. So what is gene therapy? Gene therapy is a medical treatment that involves modifying the genes of a patient's cells to treat or even cure a disease. So the process of gene therapy here is that the healthy genes will be created and would then be delivered to the patient's cells by carriers that are called vectors. So gene therapy are either conducted in vivo or ex vivo. So in this figure right here, it shows the process of in vivo in gene therapy, and there are only two vectors that are called non-viral delivery system and viral delivery system. So what's the issue here? One reason why gene therapy cases fail is because the delivery of the genes, specifically the failure of targeting the wrong tissues, which may cause the patient to have even more health problems. So the current state of gene therapy in the industry is that in recent years, the most common microscopy technique is used to understand the delivery of gene therapy that is called fluorescence microscopy. So that would mean that the fluorescent proteins can be used to track the delivery of gene therapy in several ways. So one of the ways is that it to attach a fluorescent protein to the gene therapy vector, which is basically the carrier. So when the vector delivers the gene to the cell, the fluorescent protein will be expressed and emit light tracking its movement.
Now I will be further discussing the process of cryo-electron microscopy. The picture on the right here is an example of how cryo-EM works, and this summarizes all the steps that I will soon be talking about. So our first step is to prepare our sample. And so what we want to do is we want to purify the macromolecule or complex of interest using various biochemical and biophysical methods to optimize the concentration of our sample. Our second step is grid preparation. And so what we want to do is apply small amount of hydrophilic substance such as carbon film to the grid. This helps in spreading the sample and prevent it from forming it into clusters. And so our third step is freezing. The grid is plunged into cryogen rapidly until it forms into glass-like ice layer around the sample. The fast freezing is essential to prevent ice crystals from forming, which can damage the sample. The fourth step is to collect data, and the frozen grid with the sample is then transferred into a cryo-EM for data collection. The microscope operates at cryogenic temperature to maintain the sample at frozen state. The grid will now be located into the microscope, and an electron beam is directed at it. The interaction between the same and electron beam will be projected and forms an image. The fifth step is to record the image by projection images is recorded using a detector such as charge coupled device, also known as CCD, camera, or direct electron detector. The detector will capture the patterns of electrons that have passed through the sample, producing a 2D projection image. Different image will be captured from different angle. And the last step is to process the image. 3D construction is where algorithms and computational methods are employed to determine the 3D structure of the sample. The 3D density map is interpreted by fitting known atomic models or building new models based on the density.
This is what a cryo-electron microscope looks like. Okay, so after knowing about the process of cryo-EM and also the issue that Lorraine mentioned earlier, our group formed a solution, and that is to use cryo-EM instead of fluorescence microscopy to monitor the delivery of the healthy genes in the vectors, so that researchers can image the structures of these vectors and then make changes and figure out a way to hopefully make the gene therapy process more efficient. So a little bit about the viral and non-viral vectors. So viral vectors are pretty much modified harmless viruses that carry the genes. Examples include retroviruses, which integrate genetic material into a chromosome and then it will be inserted into the human cell, or adenoviruses, which are shown in the figure here on the right, which introduce their DNA into the nucleus of the cell. Meanwhile, non-viral vectors are chemical-based vectors, such as lipid-based vectors that encapsulate and protect the DNA, and then it is engineered to fuse to the cell membrane and then releasing the healthy gene, or polymer-based vectors that form complexes that protect the DNA, also known as polyplexes. And this is the example of the adenovirus that I mentioned earlier. So you might be asking, why should we use cryo-EM? So cryo-EM has a higher atomic resolution compared to fluorescence microscopy. Cryo-EM has approximately 2 to 3 angstroms of atomic resolution. Meanwhile, fluorescence microscopy has about 400 to 700 nanometers of atomic resolution, and this can obviously be very beneficial to the researchers because they're able to zoom in more and analyze more detailed images. And something that Kayleen mentioned earlier, which is the most unique part of cryo-EM, is the freezing mechanism. And because the structure of the carrier will be freezed, the structure is preserved. Hence, when the researchers are observing the structure, it's unlikely that the structure will be altered or denatured, hence making the structure more accurate and easy to analyze. And also, for some background context, we have researched on past studies that involve cryo-EM in observing gene therapy carriers, and it's one of which that we search is actually about the adenovirus that we displayed here. And in that study, at first researchers thought that all species of adenoviruses have the same ring-like structure. However, when they put it under a cryo-electron microscope, they realized that the surface is different, and that surface was actually would determine the receptor that they would target the human cells to. Hence, it's obviously very important in knowing that surface, and now they know how to differentiate between the 2 different species of adenoviruses. So that was really helpful. So with future directions, like I mentioned earlier, with more knowledge on the structures of these carriers, it would definitely hopefully enhance the efficiency of gene therapy when the researchers are able to modify the structures of these viruses. And obviously, while cryo-EM has their benefits, our group recognizes that they have certain drawbacks that fluorescence microscopy can overcome, such as the fact that the organelles can emit light and be easier to track. And so maybe a future plan would be to incorporate both fluorescence and cryo-electron microscopy together. So maybe not simultaneously, but both methods can be used to analyze these carriers so that we can take advantage of all of the benefits within these 2 methods.
And these are our references that we cited. And here is a contribution by our team members for today's presentation. Thank you for listening.
Questions from our audience, please.
have you considered using scanning probe microscopy to directly try and have a receptor react to the gene therapy, or?
Yeah, we thought someone might ask that, so I was already researching onto scanning probe as well. And so when we initially thought of a project related to cryo-electron, we were definitely more interested in the freezing mechanism, and I feel like that, like when the fact that the freezing mechanism can sort of reassure us that the structure won't change, like that was the part that we were very interested in and we thought would be beneficial. Obviously, scanning probe has their own advantages, like being able to scan the surface, and I think the atomic resolution might be even higher, but I think an adenovirus is just like a couple of nanometers, like 100 maybe, I'm not sure, but I feel like the cryo-EM atomic resolution is already enough, and we just wanted to emphasize the freezing mechanism that we think is the most unique among all the other microscopy techniques, and yeah, that was what we wanted to show in our project idea. Thanks.
Other questions?
So normally with cryo-EM, right, since they're frozen, the particles aren't moving a whole lot. So I guess my question is, like, would you want to be tracking like the 3D structure more, or are you trying to find the 4D, like how it actually binds?
I think we're focusing more on the structure of the carriers, because that was the part that we were focusing on, because I feel like with fluorescence, like I mentioned earlier, fluorescence is more of in charge of like the delivery and like the movement, obviously because it can emit light. So I say we're more suggesting on using cryo-EM on like the structure. Like I said with the previous study, when they studied the different species of adenoviruses and they were able to detect the difference in the texture, we're proposing sort of like a similar thing. Maybe an idea would be like using non-viral vectors that are not viruses that we mentioned earlier, like lipid-based vectors and polymer-based vectors.
Any last questions? Okay, great. Thank you. Okay, group 7.
Hello, everyone. I am Spencer Foss, and I am currently going to the San Diego Jewish Academy.
I'm Mallory Prevent, and I go to Palisades Charter High School.
And I'm Wendell Wang. I go to the Kincaid High School. Our project is about examining molecular nanomachine interactions with cellular membranes using high-speed atomic force microscopy.
So nanoscale machines are an emerging technology that can directly interact with cells at a molecular level. Because of their small size and rapid movement, imaging their effects in real time is difficult.
However, we can use a newer technique of scanning probe microscopy called high-speed atomic force microscopy, and that way we can actually study the direct interaction between the nanomachine and the cellular membrane in real time.
We concluded that the application of atomic force microscopy would be a significant improvement on the state of research in this topic because of its molecular resolution combined with its usability with live samples. So synthetic nanoparticles have been a vital part of modern medical treatments with proven applications in drug delivery, immunosuppression, and hormone control on the cellular level. They have been used for contrast in microscopic imaging. They've been used to carry drugs and genetic material on the cellular level. So nanomachines are defined as a type of molecule that can produce mechanical movements in response to stimuli. For example, the contact with UV light and the introduction of chemicals or physical contact with another molecule. Natural nanomachines such as ribosomes and kinesins, which you see animation of there, regulate cell functions such as mitosis and production of DNA in living organisms. Artificial nanomachines are an active area of research in biotechnology, and work is being done on examining their potential applications in medicine. For example, some medical researchers are looking into their use as antibiotic agents, and they're also being discussed for their potential in cancer therapy.
So, as mentioned before, an area of increasing importance in the field of nanotechnology is the fabrication of mechanical devices built from molecules. Nanoscale versions of many common mechanisms have been made, including motors. These so-called molecular nanomachines, or M&Ms, are light-activated rotors composed of two identical halves connected by a carbon double bond axle. Illumination by UV light causes the carbon double bond to break and reform in a way that rotates one half 180 degrees relative to the other, effectively spinning the motor. And this is a conformational change that is reversible, allowing the motor to continuously rotate under UV light. And if you can see here, this is just a diagram of them spinning. See, one part here spins around, and then there's a conformational change, and then it spins again. And this is just the application of UV light, and this is just an overhead view of that. So M&Ms have a multitude of possible uses, but one recent application with a lot of potential is the puncturing of cell membranes to cause lysis and cell death. The rotation of molecular motors is fast enough to bore holes through membranes, and they can be combined with peptides to target specific cells. This application was presented and tested in the 2020 paper, Molecular Nanomachines Can Destroy Tissue or Kill Multicellular Eukaryotes, which proposed the use of M&Ms against diseases like cancer or parasite infections. And so this is just a sample image of the M&M boring through the cell membrane and causing lysis.
This first paper, co-authored by Professor Jeff Miller, the current director of the California Nanosystems Institute, provided evidence that molecular nanomachines could have viable applications in antibiotic treatment. The paper outlines the mechanism of bacteriocins, viral natural nanomachines that aid in the puncturing of bacterial cells, and suggest that they hold promise as precision antibiotics. We were given this paper by the program and wanted to look further into the field, which brought us to a similar paper concerning molecular nanomachines. Molecular nanomachines can destroy tissue or kill multicellular eukaryotes, published 2 years later, provided evidence that an artificial nanomotor of their design, activated by ultraviolet light, could cause cell death in specimens of a species of nematode, C. elegans, a plankton-sized crustacean, Daphnia, and in some lab mice.
Okay, so here we go. This is the main microscopy technique we want to use. It's called scanning probe microscopy. So the whole concept is that we are feeling rather than seeing. Now, normal optical and EM microscopies involve actually using light or photons, photons or electrons, but here we are actually using a tip, a cantilever, which is just fixed at one point and kind of free to move, and then we're also using a laser receptor right there, and then a flat surface to hold our sample. And then what's very useful for this is that we can actually get an idea of the topography of the sample, and then also image incredibly small samples down to the atomic scale. So one of the most important parts of this is that it is a feedback loop of the force caused by the probe making contact. Now specifically, this is actually a diagram of atomic force microscopy. So the atomic forces of being so close with our probe that has only one atom at the very tip moves the probe, and then this causes the cantilever to adjust and a laser pointing down to move across the receptor, and then this detector is able to figure out the topography based on those atomic forces. And then here is actually another useful application, is that we can understand the forces acting, because the cantilever using Hooke's law can actually act and kind of receive the force based on the movement. An important part, though, is that we want to keep this probe giving us the highest resolution images. So we're trying to reduce friction, and this is where a feedback loop comes into play, where you can see this moving up and down as this GIF plays. And the point is that we want to keep this cantilever with a constant force or oscillation amplitude by adjusting it, and then that way there's less force acting. That's another reason why here it is a tapping mechanism, so that we reduce the friction, because friction can lower our resolution. And then here is the specific type of scanning probe microscopy we want to use, high-speed AFM. Here is a GIF of what it's been able to do, which is a myosin fiber right here walking across an actin filament in real time. This is amazing because it's an incredibly small molecule, and I believe it might be a type of molecular image. But yes, so what we actually really want to study is the interaction between our M&M spinning motor going straight into a cellular membrane, and we want to study the interaction between the cellular membrane and the M&M. So what's important about high-speed AFM is that it actually is able to go so fast rather than normal scanning probe microscopy, which is considered one of the slowest forms of microscopy, because of hydroplaning. This may have happened to your car before when it gets wet, where you kind of lose friction and you just start sliding. So the actual tip in high-speed AFM, because we're going through a sample in a liquid environment, the tip goes so fast that it just starts to skid. And then because there is no friction, we don't need to use a feedback loop and adjust the cantilever. And then that way we can just constantly slide. And then another thing is the improved parts. I think I forgot to mention the raster scan. But basically with normal scanning probe microscopy and AFM, you have to just go back and forth slowly, pixel by pixel. But with an improved raster scan and this hydroplaning effect with lower friction, you can just slide, and get an image in really fast time and in very high detail.
One challenge we noticed in the literature that we looked at is of imaging the effect of nanomachines on the cell membrane is the interaction at the cellular membrane has been difficult to image with confocal microscopy due to its molecular scale, and with electron microscopy due to the difficulty of imaging live samples using techniques such as cryo-electron microscopy. Using high-speed atomic force microscopy could allow for molecular resolution imaging of a moving nanomachine while it's working, rather than looking at it after its effects have already happened.
So here is a possible experiment that we could actually use for what we've been talking about with the interaction with the cellular membrane. So this possible experiment would be involving the actual optimal UV time to activate these motors, because UV is a very high power energy, and this specific light is actually from the research paper involving the killing with the nanomachines. They found that because of the UV light, it actually harmed some of their samples, and it was at 365 nanometer wavelength. So we would like to use a sample of a human cell, because that could become cancerous. And the whole point of this experiment that we could be using with the interaction of the cellular membrane is to determine the actual optimal time to have the UV light on to cause enough damage to cause the cell death. That way we could reduce the damages caused by the UV light activating the nanomachine, but also still have enough damage to kill the cancerous cell or possible eukaryotic target.
We believe that the use of atomic force microscopy could lead to precise monitoring of individual nanomachines in accurate characterization of the interaction between nanomotors and the cell membranes they're interacting with in real time, and more precise targeting of specific eukaryotic cells. You're good.
All right, so these are our references, and we would like to give special thanks to Dr. Stieg for providing crucial information for scanning probe microscopy and AFM and high-speed AFM, I would also like to thank the CNSI staff in general. We wouldn't have done it without your support and guidance. And also, this was a really cool experience, and we learned a lot. So, yeah.
Great job, guys. Any questions from our audience?
Hello. I enjoyed this presentation. Thank you guys for doing all your research. It was really interesting to learn more about it. There was one concern or question I had on your proposal. So I was a little bit confused when you introduced the concept of UV light, because I thought that your question was about how the M&Ms would be causing the cell death. So are you trying to get the UV to cause the cell death, or is it still going to be the M&M particles?
So the UV does not cause a cell death. The UV just activates the M&M molecular motors, like they're activated. So this is just a possible experimental design, but part of having an experiment is having a control and just having different, changing a variable. So in this possible experiment, the independent variable would be changing the UV time to how long it needs to be exposed to tear through the cell membrane, and we'd at the same time be using scanning probe microscopy to observe their function. So seeing how the length of UV exposure impacts how they rotate and bore through cell membranes. But this is just one experimental design. It's part of the larger question.
Hopping in here as well, the UV is not what actually is killing the cellular membrane. The UV activates the nanomachine that spins into the cellular membrane. The UV light was found to cause other detrimental effects, and we would like to use this possible experiment to figure out a way to reduce the damage by UV to non-target cells, and that way the nanomachine could have less detrimental effects to a live sample.
Other questions?
Well, Rita's really far away now, so.
Thanks for the presentation. I really like the fact that you took the stuff you heard from Jeff Miller and then identified a different sort of direction and really pushed it a bit. Of course, I also like it because you're talking about AFM, and normally we don't get a lot of presentations that include AFM because you learned about it yesterday. So I'll also give you guys props for digging so much into a technology that you learned about yesterday. I know we talked yesterday about the fact that we've been able to visualize rotation of molecular motors right before. I wonder where you see the difference between seeing a molecule and seeing a cell, right? Even though the AFM can do both, right? They're two very different experiments, as you saw in the lab tour yesterday. I wonder if you could just think about any challenges that you foresee, the difference between visualizing a motor spinning versus seeing that molecule moving towards a cell while spinning, right? And then doing what you hope to understand. What might be the challenges there?
Well, one issue is that while the motor was spinning, we will be, tapping with a scanning probe. So there is the issue of disrupting the motion. So we have to, again, part of the high speed FM kind of just moves along. So the tapping might not be as bad, but we still may have to overcome making sure we're not messing with the motion. The other issue is that we have to image it interacting with the cellular membranes, and since we can't really use it's kind of hard to use AFM on an actual cell, so we probably just take out a section of the membrane and image it. It's kind of a bit of a finicky process, but the technology is there, and I think this proposal could definitely happen.
One more thing with the high-speed AFM. So normal scanning probe microscopy normally involves a very, very sharp tip that may puncture and damage cells. However, we learned yesterday that there can be specialized tips better for working with cells that are more spherical, so it's less pressure applied. That way we could still scan the cell. As Ellery mentioned earlier, it may be harder to scan the spinning because there could be some sort of interaction that throws off the spinning. However, I think that if we use a partial part of the cell, we should still be able to get some form of result that would be beneficial to understand the interaction with the motors.
Okay, great. Thank you.
you so much for listening.
Okay, group 8.
Microphone, laser pointer.
Hello everyone, welcome to our presentation, and today we'll be talking about lithium-ion batteries and solar panels. I'm Mina.
I'm Chloe.
I'm Leah.
So, this is an overview of what we're going to talk about today. And our research question is, how can scientists use lithium batteries to make solar energy more accessible and transition our planet into more sustainable energy sources?
Our goal is to deeply investigate the operation and degradation of batteries connected to solar panels over time. We are particularly interested in understanding the impact of variables such as temperature fluctuations, frequency of battery usage, and power consumption on battery life. It is relevant to today's modern issues of climate change as researchers are searching for more sustainable energy resources. We are approaching this issue by closely researching lithium batteries using electron microscopy. If researchers can discover which make battery limited in capacity, degrade, and unstable, they can potentially reverse these factors, which can significantly impact our modern world by making solar energy more compatible and allowing people to be more dependent on solar energy or sustainable energy rather than fossil fuels.
This is a structure of a solar panel, and it is made out of aluminum frame, and then there's tempered glass, and then there's the EVAs here, and then the main part is the solar panel, like right here, and there's a backsheet and a junction box. And then when the sun shines onto the solar cells, the energy goes to the junction box here, and then it transfers it to the battery, and it is being stored as a DC electricity. And then later, the DC then leaves the battery and enters the inverter to be converted into an AC electricity that homes can use for electricity.
This is the structure of the battery. The cathode is where, this is the cathode, and it's where the reactions that reduce the discharge occur. And then over here is the anode, where the oxidation reactions take place. And then there's a separator in between them that prevents short circuiting or like direct contact between them. And then lastly, there's the electrolyte, which is a medium that facilitates movement of lithium ions between these things. So we're going to be talking about electron microscopy, which is a technique that involves using a beam of electrons to magnify the object's image. And there are two main types. The first type is transmission EM, which is used to examine small and thin samples, and the electron beam is able to pass through the sample. And then there's scanning EM, which is used to create detailed images of the cell surfaces, and it does not damage the sample. And then in this image, you can see how SEM is just hitting the surface of it, and then TEM is able to go through it. And then TEM is able to get images like of inside the cell, while SEM is only able to get like detailed images of the surface. And then our scientific field is engineering, because our research can contribute to battery research and making better batteries.
Now I'm going to talk about the background information for solar panels. Nanoscience has facilitated the development of advanced photovoltaic materials for solar panels. Nanoscale structures and engineered interfaces and materials like silicon and quantum dots allow for enhanced light absorption and improved charge separation and reduced energy losses. These developments lead to higher conversion efficiencies and improved performance of solar panels. And the next one is transparent conductive films. Nanoscience has revolutionized the development of transparent conductive films used in solar panels. Materials like indium tin oxide and graphene-based nanomaterials offer excellent electrical conductivity while maintaining high optical transparency for the sun to come down to the solar panels. These films are essential for collecting and extracting electrical charge from solar panels, enabling efficient power conversion. Now I'm going to talk about batteries. Electrode materials are something in the battery that's like really significant, and batteries consist of advanced electrode materials with enhanced electrochemical properties. By changing the structure and composition of the electrode materials at the nanoscale, researchers can improve the battery's energy density and stability. Using nanomaterials such as nanowires and nanotubes, it can improve electron transport, which leads to better battery performance. And electrolytes are also a crucial role, which helps ions move between electrodes in a battery. This helps with ion conductivity and reduce internal resistance in the battery. This can also help improve battery health and overall performance. We are proposing to use electron microscopy to closely examine lithium-ion batteries and their materials to find out what is causing them to degrade, have limited capacity, and to be unstable. We specifically want to focus on using EM to power solar panels, optimize their efficiency, and make them able to work in various weather conditions.
How does it work? So first we want to examine NCA particles, which are nickel, cobalt, aluminum oxide, and it's a cathode material for the lithium ions. And we're going to use scanning electron microscopy to view the surface of the particles. And then we're going to use transmission EM to view more in detail, like inside the particles. And we can just closely analyze the batteries and their structures to see what we can do to make them more efficient.
You may asking, why is this better? So it is better because unlike fossil fuels and other energy sources, solar energy production is less predictable. It can fluctuate seasonally and even hour to hour as local weather changes. Moreover, researchers can obtain high-resolution images of battery materials, examine microstructural changes, and study nanoscale crystal structures. This allows detailed analysis of battery components and their behavior. The proposed project raises several questions and open doors for further discussion and research. Some potential future research include advanced materials, research and development of advanced electrode materials and electrolytes with enhanced electrochemical performance. This could involve exploring nanoscale structures such as nanowires and nanotube to improve electron transport and ionic conductivity, thereby enhancing battery performance and lifetime. Our project also raises questions about the discussion of creating more efficient batteries that can store energy and help transition our planet into using more sustainable energy resources. Many people argue that lithium-ion batteries are unstable and have the possibility of catching fire. Our proposed research can help ensure these batteries are stable and more efficient, and specifically can be used to store energy collected from solar panels. Solar panels collected energy from the sun and can be used to power things. However, when there is not enough sun to be collected, such as at night, we still need energy. Using lithium-ion batteries that can store higher capacities of energy and last longer can help address this concern, as we can store the energy in the batteries to be used when there is no sun.
These are the credits and our works cited, and then thank you for listening.
All right, questions from the audience, please.
I really liked your presentation. I just have one thing to ask. So I understand you're looking into the thing using SEM and TM. Is there like anything specifically that you're looking for?
We're specifically looking for the microstructure of battery materials at the nanoscale level. Like, this is crucial for understanding the intricate details of battery operation and performance. Do you want to add on?
We're not really looking for anything like in particular, but we're just trying to like find connections between the battery efficiency and like their structure. For example, like a previous study linked like the connectivity between the molecules with how
much it can hold. So then they were able to make more connections, which would make it have a higher capacity.
Other questions?
Why nickel, cobalt, aluminum oxide as your example?
It's a material that's used in the, like, as a cathode material, and it has a really, like, important factor in the battery's capabilities. So by examining it, we can, like, try to find connections between its structure and how the battery works.
Is it one of, like, the really emerging cathode materials? I don't know. It is? Okay.
Any other questions? Dr. Rita Blake is going to make me walk all the way.
So forgive me if you kind of already answered this, because you're speaking a little bit quietly, so sometimes it's hard to hear you. But just sort of following up on the what you're looking for question. So can you, like, based on how lithium ion batteries work and the sort of intercalation process of the lithium ions going in and out of the battery, can you describe in a little bit more detail the types of things you might think you would see if you're noticing degradation in the battery?
That's a really good question. If we go back to the structure of the battery, like, the battery's ability to perform is kind of based on, like, how the electrons are able to move between the cathode and the anode. And so we can look for, like, specific things. We're not entirely sure, because we were mainly focusing on, like, the main idea of using electrons and, like, applying it to, like, everyday problems. So
So, because I think that the idea of, like, using electron microscopy to look at degradation in batteries, like, makes a lot of sense. I think intuitively, I would say that you'd probably get more degradation, or sort of see the evidence of why it would go wrong with the lithium ions themselves being unable to sort of move between the two, as opposed to the sort of external current motion in terms of, like, how the structure actually affects the function. So that might be sort of a thing to think about in terms of, like, if you're going to look at an SEM and you see that the structure looks different, like, why does that matter? Like, is that going to matter for the electrons that are sort of going externally kind of as a circuit around the battery? Or is it going to matter because the lithium ions are going to move differently based on the fact that the two structures degraded and therefore are different than they were when they started?
Okay, any last questions? Okay, thank you guys. Okay, group 9, please come up.
Hi, I'm Jason.
I'm Jack.
And I'm Axel. And our research topic is applying cryo-EM to nanomachine treatment for cancerous tumors. Here's the abstract. Operating at the nanoscale level, bacteriophages and other biological agents are programmed to target infected cells before injecting them with carried genetic materials that act as a vaccine. Because viruses are always evolving, scientists must adjust the design of these nanomachines to target and effectively treat more formidable pathogens. Cryo-electron microscopy has been developed to immobilize the phages in their natural states, allowing researchers to monitor the contraction process so that they can make improvement to the phage design. Phages plays a central role in cancer treatment, so we believe that refining cryo-electron microscopy will allow scientists to make great strides towards mitigating the harmful effects of tumors.
So next our concept and topic, which involves the type of microscopy we want to employ, and then the scientific fields that it pertains to. So cryo-electron microscopy permits examination of live specimens at the nanoscale level, and it utilizes a sort of paralysis or immobilization to retain the natural physical or chemical form of each sample. And then the scientific fields that we can apply cryo to is oncology, based on its ability to immobilize cancerous tumors during development, and pharmacology, because it can present a coherent image of the phages that we need to treat various infections and other diseases. And moving on to the precedents, specifically the scientific approach to looking at phages before the invention of the electron microscope. So light microscopy or optical microscopy was originally used to observe cancerous tumors, and these microscopes have much more limited resolution, which would render images out of focus, at least compared to the more modern electron microscope. Most images of these tumors were incomprehensible because of this, and at this point, microscopy was not advanced enough to play a significant role in cancer treatment.
Moving on to our microscopy techniques. So first one is fluorescent microscopy. It's basically utilizing photons to view samples, but by using confocal, we eliminate out-of-focus lights to create a better image. Also, fluorescent dyes or quantum dots are used to illuminate the samples, so basically we can track the chemotherapeutic agents present in the cell. Another one is cryogenic electron microscopy, which utilizes electron beams to collect the sample image by detecting the reflected electrons. CEM uses frozen sample that is frozen with liquid nitrogen, which is around 77 Kelvin, in a vacuum state, which allows these samples to be preserved in their native structure without breaking them. This method provides information on a subcellular level and can be reconstructed in a 3D model.
All right, so that right there are R-type biosins. So exactly what are biosins? So biosins are used by bacteria to kill their competitors for the cells they're infecting. So with this biosin, we can like adapt their structure to like build these chemotherapeutic contractile nanotubes. So these chemotherapeutic agents will be located inside this spike inside this nanotube. So basically, these biosins are made of three main parts. So one of them is a sheath, so outside, and one of them will be the spike with an iron spike on the edge here to puncture through the membrane, and of course the base plate where the nanotube itself lands on the cell. So these nanotubes should be designed to encapsulate these chemotherapeutic agents, because nanotubes here, they're like 2 nanometers wide, so not any substances are like able to go inside those nanotubes that are those thin. So they must be designed to encapsulate these agents by changing the charges and make sure they stick to each other. And besides the nanotubes to be designed, the tail fibers on the edge here, you can see those tail fibers should be designed also to activate on sites of cancer. So they won't, to limit the amount of off-site injections, they should be designed to only take effect on cancerous cells. So this is basically the process of the chemotherapeutic contractile nanotubes. So first, there will be the pre-contraction nanotube, and it will attach to this membrane, and it will inject. So the sheath will contract, and this spike thing will be pushed down into the membrane. And you can see this edge going out, so those agents can be released inside the cell. So first of all, before I talk about like why it's better than like general and conventional chemotherapy, we should talk about the limitations first. So it's quite a challenge to make things this small and that powerful. So there may be some limitations and problems encountered during the penetration of the spear and the tumor. So because tumors are like hard to penetrate, and like with this single atom iron spear, it might have problems coming through. So another limitation I talked about with the biomarkers, there may be off-target injections. So these things may inject healthy cells instead of cancer cells, and the cancers are hard to biomark. So there are no therapeutic biomarkers available in cancer cells right now. So yeah, maybe it's very hard to detect cancer cells. So yeah, okay. So why is this better compared to the conventional chemotherapy? So contractile nanotubes selectively target the harmful bacteria, and because of this selectiveness, these chemotherapeutic agents only target cancerous tumors and not the healthy cells. Not like the maybe conventional chemotherapy where they target the whole body because they're intravenous, and this may affect weakness, fatigue, or maybe hair loss in some cases. So okay, so yeah, to reduce the harmful side effects of conventional chemotherapy, and also these substances inside these nanotubes, they can be used to eliminate viruses that may promote development or more further development of cancer cells. So by the use of what Jason just mentioned before, quantum dots and fluorescent dyes, these can track the chemotherapeutic agents inside the cell, so you can know if they're working or not. So if you see it through the fluorescent microscope or maybe CLM, which is a mix of fluorescent microscopy and electron microscopy, you can provide 3D high definition images of these, cells being injected by nanotubes, so you can see whether they're working or not by these tracking agents.
And finally, our future directions. So cryo-EM shows a lot of promise in a variety of different scientific fields, but we think we can apply it particularly to renovating cancer research. So we think that it has such potential because it has higher resolution than most forms of optical microscopy. And using the cryogenic temperatures that are necessary for this technique make photo bleaching and out-of-focus images more unlikely. The high pressure that is also necessary to be applied to the specimens keeps the structures pristine in their natural states. And this can help researchers provide insight on molecular organization of cancer cells, like the harmful antigens and proteins that need to be studied. So thank you, and any questions?
So first of all, I thought this was a really great presentation. You guys did an awesome job. So my question is, if you're already using pyosins to puncture cancer cell membranes, then since the puncturing is like ripping open their cell membrane and causing the cells to die, why do you also need like chemotherapeutic agents to be injected in if you're already just like killing it anyway?
So cancer spreads through the cell organelles. So they start with the membrane and they go through the cells, by those tracks of. I did this research on this article where cancer particles go spread into the cell through these pathways, and using these quantum dots, which are very small, maybe these cancer cells can be like these cancer particles can be tracked down when they move into the cell and how they develop. So with these chemotherapeutic agents injected into the cell, they would most likely follow the pathways that I just talked about, and not just on the membrane of the cell itself. So that thoroughly clears out and kills the cancer inside the cell, and maybe and hopefully it will make the cell healthy again.
Other questions from the audience, please.
So, kind of building on Ellery's question, would the pyrosin actually be breaking the cellular membrane or to kill the cancer, or is the injection of the chemotherapy actually the thing that kills it?
Yeah, for the nanotube to puncture and the spear to puncture, it must break the membrane. So I think it will break the membrane itself and kill the cancer cell. But maybe the hope of making the cell healthy again is not possible with that assumption. But maybe this is just a proposal. I have no idea of how this exactly works and how it will work. So yeah, that's it.
So it's kind of a potential,
Yeah.
if the stabbing of it with the pyrosin is not enough to kill the cell, you're going to have a anti-cancer agent also being delivered to either therapeutically cure the cell potentially or destroy the cell.
Yes,
Yeah.
exactly.
Just to add one thing, just if you guys want to know, that like pyosins are part of like a class of bacterial, like of bacteriocin nanomachines. There's also ones that bacteria make that I forgot I forgot what they're called, but they basically do what you're talking about. They inject into their like competitor bacteria like bactericidal agents. So if you got, I mean, even if you don't directly use pyosins, you can absolutely do this with a different thing. So yeah.
Yeah, so evolution has already kind of developed these things, and then you're trying to apply them to something different, yeah?
Yeah, so not just nanotubes are used right now, and some viral therapy is currently being used. They're called oncolytic viruses, which are like therapies that kill cancer through healthy viral infections that may target the cancer itself. So it acts similarly with these nanotubes that I was talking about, but basically, these nanotubes just enhance the function of killing and targeting with biomarkers possibly. So yeah.
I think we talked a bit about this yesterday.
Yeah.
How do you plan to get the drugs inside the tubes? Because different drugs, even just simply hydrophobic, hydrophilic, right? This is a tiny nanotube. It's not like a straw, right? Do you assume these things are just going to go inside, and then how are they going to come out, right? Because the normal puncture mechanism isn't killing the cell by puncturing the membrane. It's killing the cell by creating a pathway for an ion gradient to basically cause the cell to die. It's not the poking necessarily.
Yeah.
So I guess my question is, did you look more into what I asked you yesterday?
We did.
How are you going to get these drugs into the tubes and then ultimately get them out?
We're not going to get them out, first of all, but maybe there will be some mechanism where it's similar to the sheath mechanism where it contracts, but it may be inside the cell that expands maybe. But I have no idea how this exactly works, because I'm not a doctor like you. But like, yeah, because this worked, I think in the article where I read about C. diff bacteria, and maybe this will work in an injecting way with this chemotherapeutic agents. So like, maybe that mechanism may work, maybe it will not. Maybe we'll find another way to make it work. So like,
Can I ask a question about these pyosins versus bacteriophages, right? In bacteriophages, they also have this contractile tube with these little tail fibers, but it has a head, the bacteriophage head, and that contains genetic information, right? So in that case, those bacteriophages are able to inject their genetic material through the tube, through the contractile tube into the cell. So does that inspire anything that you might do with this injection of stuff into cells?
Yeah, of course, because bacteriophages themselves, they first of all, if you do not know what phages are, they inject genetic material that will construct more of these phages inside the living cell that will ultimately burst and release more of these phages. And maybe this injection technique and these maybe mitotic divisions may inspire these agents to replicate and spread through the cell itself. So
Excellent. Any other questions? Okay, great. Thank you. Okay, our last group, please come on up.
Hello, my name is Dulcie, this is Hannah, and that's Laura. And today we'll be talking about prions and mad cow disease. So just by a show of hand, could you raise your hand if you know what prions or mad cow disease are? Okay, so we've got quite a few people, that's great. If you don't know, that's totally fine because that's what our presentation is all about. So just sit back, relax, and enjoy our presentation.
Our presentation is divided into 3 main sections. So first we will be introducing what mad cow disease and prions actually are. Second is our solution with our research proposal. And third are our future directions where we can apply our proposal to more practical purposes.
Without further ado, let's get on to learn more about mad cow disease.
Okay, so another name for mad cow disease is bovine spongiform encephalopathy. And to sort of break that down, bovine means cow, spongiform means sponge-like in shape and consistency, and encephalopathy is a brain disease. So the pathogen in this disease is a prion, which is a modified protein structure that changes into a malevolent conformation that will infect the brain cells of cattle and kill them. So if a person makes contact with or ingests like infected cow meat, that person will also contract the fatal neural disorder. But the problem is that not that much is known about the structure of prions right now or what causes BSE. But a solution can be found with scanning probe microscopy or SPM. So essentially SPM is just more precise and provides better details of particles as small as atoms. And of the 3 microscopy techniques we learned, this has the greatest magnification potential and can allow scientists to clearly visualize each protein strand and structure. So then the question becomes, how can scientists examine the deformity of prions to learn more about mad cow disease? So there are actually 2 types of SPM. There's scanning tunneling microscopy and atomic force microscopy, which is going to be the one that we're focusing on. So atomic force microscopy, also known as AFM, is most often used in the field of biological sciences to observe and detect cell structures and their properties. So AFM uses a physical probe rather than like a beam of electrons or light to actually touch the sample. In order to examine the sample with like high subatomic precision, the probe itself is a singular atom. And the probe will basically like drag or like tap across the sample and adjust its force depending on the sample's height. So the probe is trying to maintain a constant amplitude, so it'll push like harder or softer if the sample is higher or lower. And this is what provides information about the shape of the surface of the sample.
So to provide some background on what prions exactly are, they are a type of pathogen. So think of things like viruses, bacteria, and fungi. Prions are similar in that they cause infectious diseases, although they are different because they are made up of distorted prion proteins. So there's 2 types of main prion proteins. There are PRPCs, and these are the non-infectious normal protein variety. And spontaneously, they can sometimes transform into what is called PRPSCs, and this is the infectious abnormal proteins. And one of the main distinctions between both is that PRPCs usually like to float around alone, whereas PRPSCs like to congregate together. And once they do congregate, they form what is called a prion. So the panel on the top right shows the differences between PRPCs and PRPSCs. A is supposed to be a normal PRPC, and you can see that the coils on the A is shorter than the coils on the B, which is the PRPSCs. However, not much is known about how these structural differences between the length of the coils is causing the PRPSCs to become infectious.
Okay, so the reason why the structural change in prions is still so unknown is because the infectious prion proteins and the non-infectious prion proteins actually have the same amino acid sequence. And this is abnormal because normally a disease would result from a mutation in like a nucleotide sequence, and then that would cause the amino acid sequences to differ.
As of recent, scientists have been working towards discovering the mechanisms that make pathogenic prion proteins, but for now, we know for sure that PRPCs can transform into PRPSCs and infect host cells by creating misfolded, distorted proteins. And electron microscopy has already been used to see certain crystals that have been formed by PRPSCs. We also know that certain prion proteins, known as RP-RP-Res, are actually resistant to disinfectants, which makes them harder to fight back against. So how does atomic force microscopy relate to this? Well, for starters, prions are about 35 kilodaltons in size, and AFM can magnify objects at the atomic level. So this would be the perfect tool to use to magnify such small particles. And AFM can also be used in a variety of mediums, such as water and other fluids. And this is great because prions are biological molecules that need water and other fluids to survive. And the atomic probe of the AFM might also be able to be used as a stimulus that can cause the protein to change shape into its PRPSC form. So that will bring us to our proposed solution.
The first step to our proposal is extracting a sample of the brain tissue from an infected cow. And next, the prion protein needs to be purified through a process called column chromatography. Next, an AFM sample will be prepared by binding the sample to a suitable substrate such as mica, which we learned about in Dr. Sieg's lecture. Also, the bond between multiple atomic probes and protein needs to be tested by making different functional tips. And the tips can be tested by probing the protein samples in different panels in order to find the right atom or multiple atoms that stimulate the protein's configuration into the infectious protein.
All right, so now that we looked at our proposed experiments, what does this say about mad cow disease overall, and what are the benefits and advantages of using AFM for this? So, as aforementioned, I said there were several perks of using AFM over other microscopy techniques such as light and electron microscopy. So for starters, with light microscopes, you wouldn't be able to get the magnification feature that you would have with an AFM. And like I said, proteins are very, very small. They're much smaller than a cell. So maybe in a light microscope, if you were to try this experiment, proteins would look as a single fluorescent dot, whereas with an AFM, they would look much more complex, and we could actually see their surface in much greater detail. And in this way, AFM would allow for more precise imaging with greater resolution of these highly sensitive particles. And the prion proteins, like I said before, they work best in liquids. And AFM is one of the only microscopes that you can use in liquid and still have such high magnification. And AFM does not use any destructive wave or light because it's pure physical contact between an atom and the sample. So so the biological molecule is not being harmed in any way.
And now that we have looked over our proposal, we're going to talk about how this relates to mad cow disease overall.
Okay, so if our experiment is successful, then we'll have at least one atom that can successfully act as a stimulus that causes a PRPC protein to contort into a PRP-SC protein. And if that's the case, we can replicate the experiment by using different variations of that element. So for example, we can use isotopes of the elements and see if we get different results there. But it is also possible that we may get multiple different types of atoms and elements to stimulate the contortion into the second configuration. And from there, what we can discern is if these atoms have any special trends or properties that can help us define what exactly the stimulus is, rather than just having a specific element be the stimulus. So for example, what if all of the successful atoms were grouped to metals? Or what if they all had large atomic radiuses? So with those types of properties, we can be able to discern what stimulus is causing these proteins to contort and become infectious. And that would help us because we can just remove the protein from the stimulus, and that would keep them in their normal state and prevent them from becoming infectious.
With our proposal using AFM, the structure of prions can be precisely observed, allowing us to find preventative measures before the infections can even take place. So our first application involves catching the prion before it coils and transforms into the infectious form PRPSCs. Since prions develop over a long period of time, we can use AFM to catch the mutation in its earlier stages. Using AFM will also allow us to define the activation site and movement of a prion infection, broadening the opportunity for drug discoveries. For example, this is similar to the research done on apicins mechanism of action by Dr. Miller. He was able to discover that a apicin molecule infects the host cell by latching onto it with its tail fibers, then penetrating it through the contraction of its sheath. And the same research approach can be applied to prions, enabling scientists to develop interventions that prevent prion infections as well. And these applications can be specifically targeted toward the mutation of prions that occur in BSE, and hopefully using this microscopy technique will lead to the discovery of medicinal treatments, as well as a stop to the spread of mad cow disease. And these are the sources we referenced throughout our presentation. And yeah, thank you for listening.
Okay, questions?
So first of all, props to you guys for talking about prions, which is like a really hard thing to research, and great job. So two things. First of all, in terms of you, testing things that will cause the prions to change conformation, will you say you'll just be using atoms, but will you also be using other biological molecules? Like specifically, prions, like regular prions, will change into, the other conformation when they're touched by the C ones will turn into the SC ones when they're near SC ones. So will you be testing just other biological molecules that could cause that? And two, will you be testing putting things on the the malignant form to turn it back into the regular form? Because if you could do that, that'd be useful for treating them.
Okay, so for this one, I'm going to go back to our proposal. Okay, so in our proposal for our experiment, we're just trying to get a couple of panels of different types of atoms, specifically different types of elements, to see if there's a reaction that occurs that causes the protein to change shape based on the stress that is being applied by that atom. So if we could get a biological, let's say molecule, and let's say the farthest atom of that biological molecule was the atomic tip, then hypothetically we could have some sort of biological interaction. But typically the tip is made out of a metal or a non-organic substance. So as of now, I don't know if that's possible, but it would be a very intriguing experimental design to maybe test out. As for, I'm so sorry, I forgot your other question. What did it regard?
just to respond to what you said, you absolutely can put can like the tip doesn't have to be a single atom thick. You can just like stick stick a protein on the end of it and then poke it. So you can do the first thing. My second question was, will you be doing it? Could you also do it the other way, like taking like the SC conformation of the protein and poking it with stuff to try to get it to turn back into a C conformation? Because that would be a way to directly treat it.
So based on the research that we did for our project, we weren't able to find a way to do that. Yes, I agree with you. It would be a great research and development technique to try out for treatment. But it's very similar to how cancer cells have been working recently. Like they go from the normal cell to the malignant cancerous ones. And as of yet, there's no way to prevent that development. There's only killing it. So with the prion proteins, I believe it's the same way, where they go from the C variant to the SC. So unfortunately, I don't think there is a way to reform it back into its C variation. I just think it goes from C to SC.
Other questions?
Okay, I thought it was very creative. I thought I really liked the presentation in the use of AFM here. So you are probing it with the different atoms, but I was wondering, since you're doing that, how are you going to determine that the protein actually changed?
So to answer this question, I'll again be referencing a previous slide. So with the panel A and B, there are different directions in which the prion proteins move towards, and you can see that the coils are different. So if we can see from the images that the coils measurement changes, whether it's barely by, let's say, an angstrom, even that measurement is precise to us, and we can determine that it has changed into its SC variation.
Any last questions?
You're asking two good questions, right? And so if you have a bare tip that is made of different material, right, you can push and poke and pull on things. And I think in the slides I showed yesterday, they showed the conformational change of proteins, of single 2D crystals of proteins, where they pushed them and changed the conformation. But that's a bare tip that's super sharp. And so you can then image it, right? To Ellery's question, if you stick a biological molecule on the end of the tip, and then you start to use those molecule-molecule interactions to induce this change, the resolution of the imaging that's subsequent to that, because you've now got this floppy molecule on the end of the tip, goes way down, right? But you can look at the force profile, right? Yes, so there are ways to do it, right? But this, the reason I'm just pointing this out is, you guys did a great job, right, in thinking about a way to use conformational changes that only the AFM can really do. But the devil's always in the details with all these measurements, right? It can do one thing, but sometimes that one thing precludes it from being able to do another thing that it can do, right? And so, in a lot of the presentations, as you start to develop more understanding of these techniques, you'll start to understand where the limits and advantages and disadvantages are, in particular when you try to push microscopes in different directions. And so I like all the ideas. The questions are great. But just to answer those two questions, there are two unique circumstances in the way in which that microscope is used.
Yeah, so I think just to summarize that, when you have the single atom tip, you get you get atomic detail. If your tip was something bigger, you get less resolution there, but you can learn other things like interaction energies. I think another interesting thing is maybe not putting the whole PRP-SC on the tip, but maybe different amino acid chunks, like peptide chunks of it, to see which part of the PRP-SC might be influencing that change. So you could do, you can learn stuff from your first atomic force measurement that can inform future measurements that you would do. Great job, everybody.
Okay, at this time, I'm going to invite our instructors up in case they had any closing remarks that they would like to say.
Okay, well, you guys spent 8 hours listening to me talk yesterday, so I won't talk too much. Other than to say, it was great to have students back here in person this year. It's we've been doing this program for what, like 7 or 8 years now.
Yeah, about.
And it was a bit of a bummer the last couple of years to not have such, energy infiltrating the CNSI. We spend our days here all the time doing research and focused on research and scholarship and working with postdocs and faculty, but it's great to have young people coming in asking great questions. So it's really always a pleasure in the summertime to change the game a little bit and to have you all here. So it's great to have you back, particularly for those of you who flew in from, I think the farthest away was Mumbai, we decided, right? So we had some serious effort made to come to UCLA. I hope that you all had a great time, learned a lot. At the end of the day, I don't think anyone expects that you learn or are going to remember everything that you saw here, because it was a bit of a fire hose over the last 5 days. But hopefully the takeaway from my perspective is that you left here more interested than you came in. You learned something that catalyzed some ideas and are inspiring you to continue your studies. And maybe we'll see some of you back here in a couple years when you decide where you want to go to college. Every time I walk around each year, I don't know, Laurent, I would say the same, as I bump into students that were in this class or other summer programs and say, hey, I'm here at UCLA now. And so hopefully we'll get to see some of you in the future. If not at UCLA, maybe we'll be showing some slides of the work that you do 10 years from now and highlighting how great all the inspiration that you've given us and that we've given you has turned into a reality in your future. So thanks so much to you for coming. To the parents, thanks for providing the opportunity for them to come here. I know it's not the way that I would have spent my summer when I was 17 or 18, but I think that's probably because I didn't have access to these types of things, and I certainly didn't have parents of means and of willingness to put me in these types of opportunities. So thanks to the parents. You all should thank your parents for sending you all here, and it's a really great pleasure. So I'll stop with that and just say thank you all. And to the team of people, you all thanked everybody. I, in my one hat as sort of with the Institute, will thank my colleagues, the education team, the grad students, all the folks behind the back walls, and of course, the laboratory staff that have made all of this possible for us. So thanks so much, everyone, for coming. I hope you had a great time, and we can get some lunch in a minute after Laurent has a chance to chat.
Okay, I'm going to be very brief, but I think for me, it's really refreshing. Each year I'm looking forward to that class because we've been doing that for many years as educators and scientists. And I have to tell you, just being part of your presentations here, I'm getting fresh ideas, actually. Through you, I'm looking at problems with a totally different lens. So thank you for that. And also, if you remember, one of the first slides that I presented, I think it was on Monday, so it's an eternity away, right? You remember that circle that represented the knowledge, right? So I hope you increase, I know you did, but now you can see that you increase your knowledge, and I think this is really what's exciting, right? I hope you're going to take something. I know you will. And as Adam mentioned, actually every year we get students that come back to us actually as undergrads, and they say, oh, can I volunteer in your lab? And we say, yes, of course you can. So stay in touch with us, and also stay in touch among yourselves, because I think starting your path as whatever, engineers, scientists, doctors, whatever you're going to become, lawyers, whatever, this is the beginning of your network, okay? And life is all about networks, right? So take advantage of that. And on behalf of all of us, congratulations, because I think it was pretty intense, right? You learned a lot, but hopefully it was fun, right? I think that was the point, right? To learn and have a good time. So, and thank you to all of you. Thank you to the parents. And let's move on to lunch.
Yeah, so really quick, before I forget, if those of you whose hair we imaged yesterday, did not get your images in your email, please let us know. Paul sent out all of the images of your hair. We have not decided who has the smoothest hair yet, but we will we'll look at the data and see what came back. So we did send all those out. If you didn't get it, let me know. Check your phones and let me know. Do we have a picture? Do we want to get a group picture up here? Sure. Maybe we can. Does everyone want to come up here real quick? Stand in front of the sign here, and we'll grab a picture of everybody before we head up and have some food and say our goodbyes. All right. Shorter people in the front, like we've done this before. Get up here, everybody. Get up here.
Yeah. Well, I think since we have CNSI shirts on, maybe we'll look like staff. Should we put on our masks?
All right. Rita, is this big enough? Elaine, get up here. Rita, all people who contribute to this, we can have somebody. I'll get one of the parents of my phone. Who's in the center? All right, we will get it that way. Okay. Well, get in here. Elaine, you're not allowed to hide, Elaine. Okay. We'll count it down. 3, 2, 1. All right, we're good. That should be plenty. Where are we going?
Upstairs.
Okay, everyone, that concludes our Nanoscale Microscopy 2023 final presentations. We are going to go upstairs to the 6th floor patio for our reception. So thank you all. Thank you, students. Thank you, parents. Thank you for another great year of this program. Yay!